Hello everyone, I am Bhrigu Mishra and this is week 12 of Biophotonics course. So this is the last interactive session of this course. Sorry I joined late because there is some issue with my iPad, so I couldn't able to join with it. So it anyway, so let's start this last interactive session. So again in this session also we will discuss the assignments questions given in the week 12. So you already have solved it, but we will discuss it in a little bit of detail. Okay, so let's start. So question first is optogenetics can be defined as combination of optics and genetics to control well-defined events within specific cell of a living tissue. And the statement 2 is the aim of optogenetics is to understand the neuronal behavior at a cellular and subcellular level inside the living tissue. So both the statements are correct if you have gone through the lectures. Optogenetics is basically what it is given in these two statements. Okay, so both the statements are correct. So let's discuss it in a little bit detail. So special cells in the brain and nervous system are called neurons. Okay, these neurons work together to produce all of our thoughts and behaviors, or you can say these are responsible for all of our thoughts and behavior. Okay, so now it's difficult to understand these neurons. Okay, so to understand how brain does work, how does brain how brain controls our behavior, we need to understand how neurons communicate. Okay, so that is the important thing. We need to understand how neurons communicate, then only we can get to know how brain controls our behavior, because ultimately it is the neurons who controls our behavior and thoughts. And there are techniques to do this electrical simulation, light simulation, and one of the technique to understand this behavior is optogenetics. Okay, and light is used in this technique with some tissue engineering, okay, to understand the behavior of this uh, neurons. Okay, so let's move to the next question. Why do we use light to probe the brain? It can be delivered in precisely timed pulses. That is correct. It is much slower than the natural operating speed of the brain. This is wrong. So this one is correct. Can be used to study a single neuron or a group of neurons without activating the entire brain to understand the neuronal behavior. This is the very important point, because in optogenetics we can study a single neuron without affecting the neighboring neurons, and that is why optogenetics is the important very important technique, because in other techniques, for example electrical simulation, we cannot do this. Light signals cannot distinguish between different cell types. This is wrong. Has the capacity to excite a specific cells. This is also correct. Can serve as an inhibitory and excitatory tool for a specific neurons. This is also true. Okay, so as per Wikipedia definition, controlling the activity of neurons or other cell types with light is achieved by expression of light sensitive ion channels, pumps, or enzymes specifically in the target cell, specifically in the target cells on the level of individual cells. Light activated enzymes and transcription factors allow precise control of biochemical signaling pathways. Okay, so in system neuroscience, the ability to control the activity of a genetically defined set of neurons has been used to understand their contribution to decision making sorry decision making fear memory mating addiction, all sorts of behavior, feeding and locomotion. Okay, now third question is VCHR1 can channel rhodopsin when excited by light of uh, some wavelength. It regulates the flow of something ion. Okay, so it is basically the yellow light which it use, okay, to excite the sodium ions. So let's talk a little bit about channel rhodopsin. So channel rhodopsin serve as sensory photoreceptors in unicellular green algae controlling photoaxis. What is photoaxis? That is movement in response to light. Okay, they enable light to control electrical excitability intracellular acidity, calcium influx and other cellular processes. Okay, now let's talk about this VCHR1. So VCHR1 is a redshifted CHR variant. CHR1 has different spectral properties compared with CHR1 and CHR2. Usually 555, 589 excites the light. However, neuronal expression of VCHR1 is about 3 times lower than CHR2, but nevertheless its ability to be activated upon yellow light, that is wavelength 550 nanometer, 550 nanometer yellow light illumination is a useful property that contributes to the diversity in the optogenic toolbox. Okay. Now, question 4. So, what ensures that only a specific group of neurons in the brain express the opsin gene in optogenetics experiments? So, we just discussed about promoters. So, these are promoter, okay, which ensures that only specific group of neurons in the brain express the opsin gene. Okay, so let's move to the next question. Statement 1. Optical neuroimaging is done by inserting an optical fiber directly inside the cortex. Statement 2 is optical neuroimaging can be performed on sedated animals as well. So both the statements are wrong. Okay, so as mentioned in the lecture slides, imaging needs to be performed in awake and freely, option B is correct, freely moving complex animals, example mouse. Okay, fiber optic cable is directly inserted inside the cranium of the animal and is in direct contact of the brain tissue. Okay, so question 6. A three-dimensional scan of the brain can be obtained using OCT, optical coherent tomography measurements by obtaining depth profile will give you only one dimensional image. By performing lateral scan, this will give you only three-dimensional only two-dimensional image because it scans on the xy plane and A on z plane. So this is 1D and lateral scan is 2D on z direction, and this is on xy plane. Fluorescence microscopy is also not in this OCT measurement. So the correct option is D, by performing a lateral scan as well as obtaining depth profile. So from lateral scan you will have 2D profile, that means in xy plane, and by obtaining depth profile you will have a z direction image. So this will give you the 3D scan. Okay, so correct option is C. 7. FNIRS can measure from regions deep in the cortex, regions in the hindbrain, regions near the cortical surface, any region in the brain. So if you remember in the lecture, there is image of brain is given, and it was mentioned that regions near the cortical surface is measured by using FNIRS. Okay, so the image is something like this. Like this, then have like this. Excuse me. And it's like this. Something is happening here. Then you have another boundary like this, and then again like this, and then a small strip. Okay, so this one is Subara Knoit space. These are the lights. This is the light source. This one is the optical detector. This one is the scalp, and this one is the skull. And here at the bottom we'll have brain cortex. So it's something like this. Okay, so this is brain cortex, and this area is the cortical surface. So it is probe, it is like penetrated through scalp and skull, and then measure near this cortical surface. Okay, next question is, NIRS measurements mainly detects depth profile of the brain, changes in the elemental composition of the brain cells, changes in optical density caused by hemodynamic changes. So the correct answer is option C. So NIRS, which is basically near infrared spectroscopy, uses infrared light to detect changes in the concentration of oxygenated and deoxygenated hemoglobin hemoglobin in the blood through obstacles such as skin and bone. Okay, so 9. Diffuse optical tomography is select the answer that applies the most. Okay, so diffuse optical tomography is used mainly. Diffuse optical tomography is used to look at blood flow to the tissues and to measure changes in the amount of oxygen in them. It utilizes light in the near infrared spectral region to measure the optical properties of the tissues. So basically it is diffuse optical tomography, and diffuse means it can cover all directions, forward, backward and left side directions. Okay, and based on the applications it is like a three-dimensional, and it also use near infrared spectral radiation. So obviously it is a B, three-dimensional NIRS. Okay, so last question: which of the following statements is incorrect for the 650 to 900 nm wavelength range? So characteristic hemoglobin absorption range, which is true. Relatively high degree of transparency of brain tissue, and this is also true because brain tissue doesn't absorb in 650 to 900 nanometer wavelength range. Minimal absorption for deoxygenated hemoglobin. Near IR range, this is also not a okay. It is true that it is 650 to 900 is a near IR range. So the correct option is C, which is the wrong statement. So it has a high absorption for deoxygenated hemoglobin in this range. So you have so deoxygenated hemoglobin has absorption peaks. Deoxygenated hemoglobin has absorption peaks at 416, 430, 541, 556 and 577 nanometer. Okay, and also has several or I would just say that it has, okay, just remove this, like it has high absorption. Okay, no need to write the peaks. It has high absorption in this 650 to 900 nanometer wavelength range. Okay, so yeah, that's it for today's, and this was the last interactive session of the biophotonics course. So thank you very much, and please enjoy and do further reading, because this is a very interesting topic, biophotonics. And in future, if you will see lot of techniques which will help to cure our tissues for severe diseases. Okay, thank you.